Salam and hello everyone. Our group will be present video practical test of hemoglobin estimation by using Sahli'S method. Our group consists of Aimi Khafidah binti Zulkifli, Alina Jiha binti Ramli, Amira Ashikin binti Muhammad Rosli, Khadijah Alis Afifah binti Muhammad Imran, and Nur Faiza binti Muhammad Hasan. Me, Amira Ashikin binti Muhammad Rosli, will present introduction. For introduction, hemoglobin is the major constituent of the red cell cytoplasm, accounting for approximately 90% of the dried weight of mature cell. It comprises of heme and globin. Hemoglobin carries oxygen from the lung to tissue cells and carbon dioxide from the cell to the lung. After normal lifespan of RBC, the red cells are destroyed and the components of the hemoglobin undergo metabolic degeneration. Hemoglobin estimation is used to determine the presence and severity of anemia, screening for polycythemia, to assess response to specific therapy in anemia, estimation of red cell indices, and selection of blood donors. Sahli'S acid hematine enzyme methemoglobin are the most commonly used method.
Assalamualaikum warahmatullahi wabarakatuh. My name is Aimi Khafidah binti Zulkifli and I will explain about the principle of hemoglobin estimation by using Sahli'S method and the objective of this experiment. We go on with the principle of this experiment first. So the blood is mixed and by 10 hydrochloric acid so that hemoglobin is converted to acid hematine, which is brown in color. The mixture is then diluted with water until the brown color of the diluted blood sample matches the brown glass standard of the comparator box. The concentration of Hb is read directly from the scale of hemometer tube. Next, the objective of this experiment. We have two objectives. The first one is to perform hemoglobin estimation using Sahli'S acid hematine or can methemoglobin method. And the next one is to determine hemoglobin concentration in a given sample via Sahli'S acid hematine or can methemoglobin method.
Hi, I'm Khadijah and I will explain about method and methodology. The equipment and materials needed for this experiment are Sahli'S hemoglobin miometer set, pasture pipettes, and by 10 hydrochloric acid HCl, distilled water, and tissue. As for our specimen, we use venous blood withdrawn in EDTA tube. Now we begin with the procedure. With the help of dropper, N by 10 HCl was filled in hemoglobinometer tube up to its lowest marking, which means up to 2 gram percentage yellow marking. The hemoglobinometer tube then was put in a comparator box. As can be seen in the picture, the blood was drawn in hemoglobinometer tube. The content was mixed thoroughly and the hemoglobinometer tube was put in a comparator box. Then, we waited for 10 minutes. During this time, hemoglobin was converted to acid hematine, which looks brown in color. If less than the required amount of time, which is 10 minutes, then it may not be enough to convert all the hemoglobin which presents in 20 microliter of blood to acid hematine, hence giving lower values for hemoglobin. After 10 minutes, distilled water drop was added till the color of the solution is same to that of the comparator box color strip. The distilled water was kept added and the solution was mixed with the help of stirrer. The color was checked frequently. When color of the solution was same as the comparator box color strip, the tube was held, diluted, and the readings in hemoglobin gram percentage was read as can be seen in the right picture.
That's all for my part, method and methodology. I'll pass on to the next person. Thank you.
Assalamualaikum and hi. Today I am Alia Najha and I will explain to you the result of hemoglobin estimation by using Sahli'S method. So let's discover the result together. When hydrochloric acid was mixed with the blood in hemoglobinometer tube, hemoglobin is converted to the acid hematine looks brown color. If less time taken, it may not be enough to convert all the hemoglobin to acid hematine. And this will indicate a low value of hemoglobin result, which lead to anemia, and that cause symptoms like fatigue and trouble breathing. We could indicate the range of low level of hemoglobin by using this reference. For male, the range is less than 13.5 grams of hemoglobin per deciliter, and for female is less than 12 grams of hemoglobin per deciliter. Next, when distilled water was added, the color solution will be mixed. It should look the same as the comparator box color strip. Compare it at eye level and make sure the solution is brownish like the comparator box. Then, lower meniscus need to be read in hemoglobin gram% in the Sahli'S hemometer tube. The best result should be in a normal range of hemoglobin. For baby, it is 23 grams of hemoglobin per deciliter because RBC count is small. For male, it is within 13.5 to 17.5 grams of hemoglobin per deciliter, and for female, it is within 12 to 15 grams of hemoglobin per deciliter.
Hello everyone, I'm Faiza. I will explain to you guys about the discussion part from this experiment. The estimation of hemoglobin in the blood is commonly prescribed in suspected anemia, which can be caused by various factors. Nowadays, in many laboratories, the hemoglobin estimation is done by using automatic hematology analyzers. But still, in many other labs, the following method is commonly used to determine the hemoglobin concentration in a patient's blood. A female student was used in this experiment as a sample, and the result obtained is 12.6 grams per deciliter, which is in normal range. These are based on the estimation of hemoglobin by measurement of its color. The differences in results can happen due to input errors of Sahli'S method of hemoglobin estimation handling. Now, let's look at the advantage and disadvantage of this Sahli'S method. For advantages, it is easy and quick to perform, inexpensive, can be used as a bedside procedure, and it does not require technical expertise. For the disadvantages, the visual intensity may be different for different individuals. This method can only estimate oxyhemoglobin. The carboxy hemoglobin, metahemoglobin, and sulfamyoglobin cannot be estimated because they are not converted into acid hematine. The endpoint disappears soon, so it is difficult to know the actual endpoint, and also the proper stable standard is not available. The resulting solution is not clear solution, but a suspension due to action of hydrochloric acid and the proteins and lipids. The envelope of color is low and acid hematine is not stable. Source of lights will influence the comparison of colors. For maximum color, longer time is required. Hemoglobin estimation gives a brief idea of the pathological condition to the physician so that your physician can easily understand the cause of pathology and prescribe an effective treatment for it. The high level of hemoglobin can be caused by heavy smoking, methemoglobinemia, elevated erythropoietin level, for example tumor of kidney, liver, CNS, and ovary, and also renal disease, for example hydronephrosis and vascular impairment. Adrenal hypocortism, therapeutic androgens, dehydrations, for example water deprivation, vomiting, and diarrhea, and lastly plasma loss, for example burn. The low level of hemoglobin can be caused by leukemia, tuberculosis, iron deficiency anemia, parasitic infection, severely in hookworm infection, sickle cell anemia, thalassemia, aplastic anemia, hemolytic anemia, and lastly, loss of blood.
I will represent the last part, which is conclusion. As the conclusion, hemoglobin estimation is frequently performed screening hematological tests for laboratory service. Sahli'S is usually practiced because it's easy to perform, inexpensive and can be used as a bedside procedure. However, the result is less accurate. Extra precaution must be taken to get a precise result. So the normal range of hemoglobin for males is 13.5 to 17.5 gram per deciliter, while for females is 12 to 15.5 gram per deciliter. I think that's all from us. Thank you.